This procedure allows mosquito researchers to easily address their mosquitoes' reproductive fitness at the individual level. Compared to conventional methods, this protocol significantly reduces the time, space, and labor required to test mosquitoes' fecundity and fertility at the individual level.
Begin by preparing plates for the oviposition experiment. Drill 4 to 6 holes per well in the lid of a 24-well culture plate using a household drill with a 1.6 mm bit. One day before the experiment, wash and rinse the plates and soak them in 1 to 5% bleach for 30 to 60 minutes at room temperature. Then, rinse the plates thoroughly under running deionized water and dry them. Melt 2% agarose in deionized water, and immediately add 500 microliters of the molten agarose to each well of a 24-well plate. Leave the plates on the lab bench overnight to dry out any condensation. At least 16 hours before the blood feeding, remove any source of water or sugar from the female mosquitoes. On the day of the feeding, heat a water circulator to 37 degrees Celsius and feed the mosquitoes with vertebrate blood placed in artificial feeders for 15 to 30 minutes. Then, transfer the mosquitoes to a glass dish on ice. Select the ones that are engorged with blood and place them into a container with 30% sucrose water. Allow at least 72 hours for the females to finish excretion and egg development.